morning, everybody. Okay, so let's kick off. Welcome everybody to Structure Meets Function 43, the Instruct webinar, which is every few weeks. So, voilà. Okay. So Instruct, just for people who are not fully aware, this is the European Structural Biology Infrastructure. And today we're going to hear from two of the centres. There is the new German centre, which comprises several distinct facilities, and Manfred Weiss from Berlin will give us a presentation of what is now on offer from Germany. And then we will hear from Judith, who is at the ALBA Synchrotron. The partner Spain has been a member of Instruct for a long time, but has recently increased its offer through the addition of some new facilities. And Judith will also present what is now available in Spain. Look at the next slide. John, yeah. So currently Instruct has 17 member states. Or member organizations, because this also includes the MBL. These pay an annual subscription, and this allows scientists from these member organizations to access the services available at the different centers of Instruct. These services are not just research visits, although of course research visits is a main activity of Instruct, but also Instruct organizes and finances training in established and new techniques. It funds longer-term research visits that we call internships. These can last several months. And also there is distinct R&D funding for projects, which you can apply to via the Instruct website. Next slide, please. So I just give you a reminder that in May, end of May, there will be the Instruct Biennial Structural Biology Conference, following on from Portugal, where it was last time. This has become really a fantastic international high-level conference, and I encourage you all to register so you can get more information from the Instruct website. There's currently an abstract submission if you want to propose something for an oral presentation. There are fellowships available for typically PhD students, so more information is on the website there, but this will offset some of the cost of a student attending. And accommodation has been negotiated at very favourable rates for Instruct participants. And as a last point, we're still looking for sponsors. So if any of you have experience with typically companies who've sponsored your national events, please get in contact and we'll be happy to take over and try and contact them for some sponsorship. John? Well, there's a nice 2D barcode, but you can get the information from the Instruct website. The Instruct website shown here, many of you know this, so this is Instruct-ERIC.org. You can send an email, the address is just below there. And I really encourage you anyway, if you haven't already, to set up an account on ARIA, because this will then put you on the mailing list and you can keep in touch with what's going on. Judith, do you want to join us?
Yeah, I hope. Thank you very much. Hopefully, I'm not going to have any problem.
I have not.
Let's share. Okay. I'm going to share a full screen, actually. Cool. Do you see my screen?
Yes, yes.
Okay, excellent.
Just put it into full put into presentation mode, please.
Could you, can you see the first slide? Okay, thank you very much. So thank you very much for Instruct for giving me the opportunity to present the new expanded, it's not new actually, but it's so expanded that it's almost new, center Instruct in Spain. And also thank you very much for the other facilities in giving me the trust to present their work and their facilities. So actually we are not coming from nowhere. We in the facilities across Spain we have been collaborating for quite a long time. First, of course, the former Instruct-ES that was formed by two centers at the same research center, CNB at CSIC in Madrid. But we had also distributed network infrastructure such as NMR, and we have a hub in Barcelona on integrated structural biology. So we are having a close relationship, and actually that has reflected in the large extension of the center, very inclusive, and with an effort to include a large scope of techniques, and we have agreed quite transversally across the facilities that the Instruct proposals are. The share of the Instruct proposals in the total access offered by BMINE is around 10%, which is, I think, is a reasonable amount. Apart, of course, people can apply to the facilities, but directly to them actually, and we have divided the instruments and services offered to Instruct in three different geographical sites, and this is easily understandable, as you can see in the map. We have a relatively large number of facilities with a variety of techniques, from X-rays, electron microscopies, NMR, and also computational resources in three different sites. And each site with different facilities, right? So I'm going to go facility by facility, classifying by the technique actually. First talking with NMR, then the electron microscopies and computational resources, and finally X-ray techniques, in which actually we have a unique technique that was not offered before in Instruct, in the portfolio of Instruct. So I should probably hide this one. Okay, so first the NMR overview. Actually, we have the NMR instruments in the three nodes, Madrid, Bilbao, and Barcelona, and actually they form already a network, a so-called ICTS, so a distributed technical and scientific infrastructure, and with some flavors in the science that they are developing. In Madrid, the LMR facility provides access to these instruments. I'm not going to go explicitly go to each instrument because it's quite long, but essentially biosolids, cryoprobe, and this is the main focus of this lab actually in Madrid. Then the second NMR facility in Bilbao has 5 high-field spectrometers, and they are dedicated to protein and glycan sample preparation with stable isotopes or chemical labeling. These are the instruments listed below. Clearly, the most important one is actually 1 gigahertz, not only 800 gigahertz, 800 megahertz spectrometer. with some instruments having automatic sample changes and different probe heads. Then at Barcelona, there's the technical centers at the University of Barcelona with instruments available. Clearly, the jewel of the crown is the Bruker 1 GHz spectrometer with cryoprobe for proton detection, and they have also one 800 MHz and one 600 MHz for carbon, nitrogen detection and proton detection, respectively. The group is specialized in intrinsically disordered proteins, IDPs, and also biologics, for instance, exploring the natural abundance formulated by monoclonal antibodies. And they have a focus with integrative structural biology together with joining with electron microscopies and X-ray techniques in Barcelona as well. So, moving to cryo-EM, actually, starting with Bilbao, they have started not something. This is a relatively new facility, something like 5 years ago, and they do have the complete workflow of the techniques in single particle analysis, cryo-electron diffraction, and cryo-clam especially. The most relevant instruments are Titan Cryos J4 and Aculos 2 Plus, the cryo-FIPSM, right? So they do assist in the whole process of preparing, verifying, and collecting with cryo-FIPSM, and finally for the cryo-TEM for data collection. Also, another facility actually at ALBA is the cryo-EM owned, managed by IBMB CSIC, but we are in close collaboration with them, and actually we do offer for Spanish users some beam time there. This is the first cryo-EM microscope in ALBA. The equipment essentially is a Glacios, a fully equipped Glacios 200 kilovolts with X field emission gun, optics, autoloader, and Falcon 4 detector. It has already been proven for a high resolution reconstruction of membrane proteins, and this actually is quite successful already. They do have a science on that, and the staff working on it they are very helpful in going through all the process. They are actually using the ALBA premises for vitrifying and for preparing the samples. Maybe the most the classic center in electron microscopy in Spain is the CMB CSIC, of course. They have been providing since 1980s, actually, providing support sample preparation, characterization, and data acquisition on cryo-electron microscopy and single particle analysis as well, tomography, macro-ED, and they have a focus in cryocorrelative light and electron microscopy, cryo-clam, in order to allow the analysis of the same samples by this Zeiss 80 scan cryoconfocal microscope and their cryo-electron microscopes. They are going to receive soon the first Titan Cryos 5, which is going to be the first one to be installed in Europe, actually. And they do have other microscopes for sample screening and other studies as well. This is, I would say, this so far has been the reference center for electron microscopy in Spain, actually. Coupled to this, and synergetically, actually, there's the image processing, the Instruct Image Processing Center, and the same center, the CMB, And actually, they do support and train in the use of image processing software. Essentially, Scipion, they are the developers of this suite, and they develop new methodological approaches. And actually, we have been ALBA has been collaborating with them as well, and also provides computational resources. And especially expert staff actually to project in order to assist in the process of the projects. An example of what they are doing is a structure-based virtual drug screening service using a Scipion Chem. Essentially, they are doing it in 2 steps. First, they are looking for druggable pockets, and then they are predicting drug binding using their developed programs. This is, I think, that these 2 centers, at the CMB, the Cryo-Electron Microscopy Center facility, and the Image Processing Center, were already in Instruct, and probably is mostly well known by the Instruct community here. Now we're moving to X-ray techniques, and essentially this is ALBA, the synchrotron light source close to Barcelona. And at ALBA, we are offering to Instruct community essentially 2 techniques and 1 service. MX, this is new. This is not new, I mean. The service is a fragment screening platform. This is not new neither. And also the cryo-soft X-ray tomography that I'm going to explain a little bit later. Regarding MX techniques, they are the classical ones: rotational, oscillation, MX, and micro MX techniques. Also jet-based serial crystallography, and soon there will be the fixed target serial crystallography, not by 2025, but 26, it's still January. And also offline serial crystallography setup for preparation, sample prep, and optimization. And we are also offering the crystallization and fragment screening platform. Time resolve will come in midterm, I would say. We are progressing on that. And regarding the crystallization and fragment screening, this is going to be progressively open to proposals. This is still incipient, but we have a complete setup of instrumentation from Mosquito and the Dragonfly robots, Formula Trix Imager, Crystal Shifter, etc. And this is offered as a pipeline, not a standalone service, but sample preparation, fragment screening is coupled to Sherlock beamline access. And these are the characteristics of the beamline. Essentially, Sherlock is a mature beamline, recently updated, and very importantly, supports fully automated or unattended data collection pipeline. And the data is collected unattended, so automatic in typically in 1 week. We're not committing to 1 week, but this is about that time that we are offering right now. And the Shira beamline is a new macrofocus beamline, new meaning that the first PDBs were just uploaded actually 2 weeks ago. And the focus is to go to 1 by 1. We are currently 4 by 4, but going down. And here the uniqueness is that it offers a very, very low background for microfocus as a microfocus beamline, because the whole end station, as you can see in the picture in the right, is fully covered, is fully enclosed in a helium environment chamber, which is recovered, the helium. And this offers optical conditions for native phasing techniques, but also for low background in micron-sized crystals. And importantly, this is fully compatible with the standard cryopins and air operation. So I mean, we can work in helium without using a special pins or cryotools. And maybe the last technique that should be explained a little bit more is the soft X-ray tomography. That was not offered in Instruct. And now, so this is a premiere, I would say, for Instruct community, although it's very well known in the scientific field of integrative structural biology. Essentially, soft X-ray tomography delivers the 3D cell structure in a typically 10 by 10 by maybe 5 micron volume at 30 nanometers resolution. And essentially is a full field transmission X-ray microscope. I'm not going to go into details. You can find them in the bibliography, etc. And the important points here is that it does not require a heavy sample preparation, no cell slicing, those cells. It's a tomography in which cells are entire because X-rays are transmissive to cells, and no chemical or fluorophore is required. It's compatible with cryo-ET grids as well, and importantly as well, this technique allows correlative studies. And actually, Mistral is relatively productive. It produces about 100 tomograms per day in typically 2 kits, and is offered in Instruct as a cryo soft X-ray tomography pipeline. We can offer that this is optional sample preparation because we know that it's not so well known this technique. Then we go to a cryo 3D SIM that is a super resolution in cryogenic conditions visible light microscope in order to find which are the interesting parts of the grid that we should scan. And then finally, we move the same grid and with the localized spots in the Mistral beamline to perform the cryo soft X-ray tomography experiment. Importantly, this cryo 3D SIM microscope correlates with the lab or say research centers confocal super resolution microscopes. So you may have a direct pipeline from your confocal microscope until nanometer to 30 nanometer resolution in 3D on the same samples. And actually, this is the pipeline, right? So this essentially first. You see here a part of the grid, and you can also see here superposed the fluorescence that the fluorescence signal that is given by the cryo 3D SIM. And in the two different, we allow 2 different fluorophores. In the future, we may allow up to 3 fluorophores. And actually, this is one of the highlights. I'm not going to explain in details, but essentially, the cryosoft X-ray tomography gives the cellular context. And then this can be correlated with other techniques, in this case, nano XRF. X-ray fluorescence to localize which are the compounds that we are interested in this case, iridium half sandwich complex, which is a potent anti-cancer, which would be replacing cisplatin, and localize it in the cellular context without slicing the cells, right? And what we are already developing with collaborators is the correlative approach between 3D SIM, the cryosoft X-ray tomography, and electron tomography. Actually, between the electron tomography and the fluorescence cell volume, there's a huge step in the volume scanned. So cryo-electron tomography, that is Mistral beamline, offers the intermediate step in order to localize and better find and hit the interesting spot in the cell, right? So here you have actually the correlative approach between the electron tomography and the soft X-ray and the cryo soft X-ray tomography as well. So going to spending too much time in that, so what I'm going not to explain further on, but this is very recent and this has been published very recently. So so some last considerations. First, is important that as is usual, but it's worth saying that the scientific evaluations of Instruct will be accepted by the different facilities across Spain. But and also that's not so obvious. Some extra funding could be available in some facilities for good projects that could not be ranked enough to have direct Instruct funding. In ALBA, we have a specific fund, so one has to please ask to the relevant staff. And then for in Bilbao, the access projects will have a 40% fee rebate. It is in case is not funded by Instruct. And for the CMB, the access fee will be at the internal cost up to 5 projects per year. Just to know that some services are progressively being open to Instruct access. This is the case for the fragment screening platform, for instance. And still, we are quite new, and some websites of the facilities are still in the Instruct website are still being updated. So in case of that, just contact directly the facilities, and we will be keen to inform you about the developments and the opportunities. And just to finish, we are inaugurating this new expanded Instruct Spain Center by having a kickoff workshop, which is going to be done at the end of this month. Registration is open. You see here the invited speakers, and so I think it's going to be a very nice workshop in order to understand and to see and to be in contact with the facilities and the managers of the facilities. And essentially, this is it. Thank you very much for your attention. Open to questions.
Okay, thank you very much, Judith. What I'll say is, please, could you just type any questions you have into the chat and not the Q&A. Let's keep it in the same place. While I'm waiting for that, I'll just open with a couple of questions. So, of course, we all know that since COVID, access has been more and more remote with less and less user visits. How do you see the balance of these two modes for your beamline access and your cryo-EM access? Do you expect users to ship samples? And I will just say Instruct contribute towards the price of the shipping, or do you expect users to come physically? COVID had a real impact clearly in how the facility, the instruments and services are being used. That's clear. We expect everything actually from users. Come on, we are running facilities, so we know that there's a lot of casuistic. That depends a lot on the state of the project. That depends on the group and depends on the technique. For MX, clearly we expect either remote and operating the beamline from host institution. If this is the case, I would say first visit could be only one shift, but first visit should be done at on-site in order to be familiar with the beamline, if not already, if you have not used it. For other X-ray techniques which are not so standard, and for other projects in NMR especially, we expect the people to come. For electron microscopy, can be done. I don't think in the beginning can be done, but most probably in the near future, some projects could be done remotely. But this is again facility dependent. Please ask the facility managers in order to know which are the specificities. Okay, and I'll follow that on for the NMR that is on offer. Is there also sample preparation, so the expression, the isotope labeling and the purification, or do you expect users to prepare the sample and ship this then for analysis?
Hard to say because I'm not an NMR person, but I would say all the facilities are prepared to do the sample preparation and to do all the labeling and all the studies are required in order to perform a successful experiment. So they are fully prepared. Whether they can offer this in or not in Instruct, this is still a matter of discussion. I think that depends on the service level agreement, and this is being discussed. So if somebody is interested, that might be explored. So please contact directly the facility managers, and they will give you the best advice in order to boost your projects as far as I can be done.
Okay. All right. Well, thank you very much. I think we'll move on. Very interesting and a real good addition to the Instruct catalog, and lots of new things which are actually new in the infrastructure. So thank you very much. Let's move on then to Manfred. Manfred's presentation is nice and visible. So let's hand over to Manfred.
All right. Thanks a lot, Darren. I hope you can all hear me. Sorry for the technical difficulties, but let's see how we get it to work now. So I'll be talking about Instruct Germany. Before we jump into Instruct, I would like to say a few words about the structural biology in Germany on the next slide. Germany is one of the largest countries in Europe, obviously, and also has a very large structural biology community. There is, let's say, more than 100, possibly 120, 130 research groups in Germany, which have a significant focus on structural biology. There is 3 large-scale facilities for X-ray crystallography. That's the DESY in Hamburg, the HZB in Berlin running the BESSY Synchrotron, and there is EU Exfeld in Hamburg or in Schenefeld. Then of the roughly 10 or 11 1.2 gigahertz NMR magnets, 4 of them are located in Germany, in the University of Frankfurt, Helmholtz-Zentrum Munich, FMP in Berlin, and the MPI in Göttingen. And there is roughly 40 cryo-EM installations, with most prominent centers being the CSSB in Hamburg, the Helmholtz Center in Jülich, then Max Planck Institute and Technical University in Munich, and the Charité in Berlin, and of course many more. Now on the next slide, I'll show you that Germany is Instruct member since 2024 only. So we joined Instruct relatively late. There was, over the years, there were several attempts which for one or the other reason did not work out. So finally we managed to become member in 2024. And then at that time, of course, there was no German Instruct Center established. So our ministry issued a call for establishing a German Instruct Center. And on the next slide, this call was published in July. We began by sending out invitations to all German structural biology groups. We received 12 letters of interest. We had a preparatory meeting, and then we prepared an application which was submitted to the ministry. This was reviewed by Instruct, by the Instruct Scientific Advisory Board, and based on the feedback from the review from the ISAB and also from the discussion at the Instruct Council meeting in May last year, we submitted a revised and consolidated proposal, and this was finally accepted by the Instruct Council in July last year. Now on the next slide, the consolidation basically meant that we had to reduce the number of sites. So initially we had 12 partners in 7 different geographic locations, and essentially we went down to 7 partners in 5 geographic locations. I'll get to the individual services in the next slide. Showing the governance structure on the next slide is so the coordination of Instruct Germany is done by Clemens Glaubitz from the University of Frankfurt. So essentially we have a distributed center, as I said already, over 5 different geographic locations: Frankfurt, Munich, Berlin, Hamburg, and Jülich. Each of these partners provides a member to the to our executive board. Then we feature different techniques, obviously NMR in Frankfurt and Munich, X-ray techniques in Berlin and Hamburg, with Hamburg being the site of the synchrotron, but also the X-FEL, cryo-EM in Hamburg and Jülich. Then in addition, we have a computational aspect similar to what you heard in Judith's talk. This is a new service to Instruct. I will talk about this in a little later, in a few slides later. And we also have some support for RNA sample preparation by the NMR group in Frankfurt. So let's go to the individual partners on the next slide. So we start in Frankfurt. We offer some liquid and solid state NMR, DNP experiments, also fragment screening by NMR. They are doing, they have quite some expertise in RNA NMR. This is also why they are supporting RNA sample production. And there is also a well-established record in European user access, Frankfurt having taken part in several of the EU access projects. The second NMR site is in Munich. Next slide. That's run by Michael Sattler. This is a joint venture by the Helmholtz-Zentrum in Munich and the Technical University in Munich. They also have a 1.2 gigahertz machine, liquid and solid state NMR. They focus more on RNA protein complexes, but also offer the full scale of biomolecular applications. Coming to the X-ray side of it, that's me in Berlin at the BESSY Synchrotron. Also with a lot of experience in transnational access. So here we offer just the general access to the 3 macromolecular crystallography beamlines that we are running in Berlin. But also we have a significant expertise in crystallographic fragment screening. We've been developing this for the better part of the past 10 years. And so this is a extremely popular user experiment nowadays at our facilities. And we also offer this in the Instruct context. The other X-ray partners are, next slide, are Johanna from DESY, bringing in serial crystallography expertise at the P11 beamline, and Hu-Yong Han, who I think is also in the room, representing the EU X-FEL side of it. This the X-FEL. This is also something that's unique within Instruct. Here I should say that the Instruct services do not entail direct access to X-FEL beam time, but help in sample preparation, and then possible also what is called the crystallization beam time. So if a sample is chattable, essentially it will very likely see the beam at some point. And then of course also the help in preparing full-fledged proposals for obtaining X-FEL beam time. Next is also in Hamburg is the CSSB with a representation of Kai Grünewald, who offers single particle and tomography access. They have a whole park of instruments there, and basically are not really limited or restrained by the type of project you bring. It just matters whether it fits in their schedule and whether they can actually support it. Next slide. We also offer cryo-EM and electron tomography at the second cryo-EM center. That's Helmholtz-Zentrum in Jülich, or Forschungszentrum Jülich, represented by Carsten Sachse. There's also a single particle and tomography access, and they have a unique setup for thicker samples. So this might be interesting for one or the other project. But in principle, they also cater to all sorts of biological problems. Jülich also offers this computational aspect, which I've been talking about. Next, please. This is Holger Gohlke. This is a Instruct pilot project, I would say, similar to what the Spanish people are offering here. Access to biomolecular informatics, this is called. This has, this ranges from simulations to coarse-grained molecular dynamics, and provides a significant number of hours of access to the Jülich supercomputers that are administered by them. So this is also on a project base. It is not just access to computing time, but it's a project-based access with scientific support by the people, and then of course also the access to the supercomputers within the frame of the projects. Okay, next slide. Let's quickly talk about the access catalog. You can already, I hope you can already find this on the Instruct websites. If not yet, then it should be on the website soon. So let's just zoom in there. Next slide. Frankfurt General Access NMR, Liquid Solid State NMR. So you can see we get 28 days on 1 gigahertz magnets, 84 and 80 days on the smaller magnets. Next slide would be Munich. Also 28 days on a 1 gigahertz NMR, and a similar number of days on the smaller magnets. I'm not mentioning again the specialties of the different groups, just the amount or the capacities they offer to the community. Then we go to X-ray. That's us in Berlin. We offer, as I mentioned, general access, 120 hours and 5 entire fragment screening campaigns. And the fragment screening campaigns obviously are not only beam time. There is some help in preparing the campaigns. This can be done, anticipating Darren's question, this can be done on site. So users can actually come to us and do all the preparation there. But we also offer what we call fragment screening to go, where we would sort of ship everything to the user lab, and users prepare all the samples in their home lab and then basically send back the samples for remote measurements. I mean, similar to other crystallography beam lines, 90% of our beam time is now carried out remotely, and this also includes the fragment screening campaigns. In addition to the preparation beam time, we also offer some help in processing and analyzing the fragment screening campaigns, and you will have access to one of our fast computing servers to analyze these campaigns, which is not a trivial exercise, I would say. Hamburg then would be next with access to the P11 beamline, mainly, well, with a specialty on serial crystallography. You can see the amount there. Then I mentioned already the EU XFEL, of the access to the biology lab infrastructure, sample preparation, characterization, support for proposal writing. The access to XFEL beam time is currently based on proposals, and we are they are discussing, or we are discussing with Instruct how this should be framed and how this will be operated in practice in the future. Then for cryo-EM, there's the general access to single particle cryo-EM and cryo-ET. There's also some specialized access, for instance, membrane proteins, correlative light and electron cryo microscopy, and also workflows for targeted cellular cryo electron tomography. And we offer 8 sessions per year covering the full workflow. And finally, there is Jülich. Again, with cryo-EM access, 75 days for single particle and 50 days for in situ biology or tomography. And then the computational methods and CPU time. So essentially, it's 120,000 hours of GPU time, which is 20% of the total available GPU hours. So this will support 5 complete projects per year, including, support software, etc. Next slide is, we had our inauguration meeting already in October last year in Frankfurt. It was a hybrid meeting. Several participants or several center representatives came to Frankfurt, but there were also a substantial fraction participating remotely. On the next slide, we are now in the state that we are open to accept proposals, although our consortium agreement is still in the making. This has more to do with the internal running of the German Instruct Center, because we are not getting any running costs from our ministry. We have to come up with these running costs ourselves. So we have to figure out how to best do this. The service level agreements with Instruct are currently being finalized, and we expect that we have final agreements in place within a month or 2. And this is also when we will start accepting users. So we expect first users in April or May this year. You can already find us on the Instruct website. And I hope that our final catalog will also be there, or is also there. Maybe Pauline can answer that if there is a question about that. Good. I think that I'm already at the end of my presentation. Thank you very much for listening. I'm open to questions. You can, of course, also email me in case you have questions about how to best get access or talk to the Instruct people. So thank you very much.
Okay, thank you, Manfred. I'll open with a couple of questions, but if anybody wants to ask anything, as again, please put it in the chat. So, Manfred, you described this. I think it's going to be one of the first possibilities to access fragment screening. So, of course, behind this there is often a theme of drug discovery and potential intellectual property. The libraries that you have, first of all, are they all available? And secondly, do they have any restrictions, or are they sort of open access?
We have several libraries available. The one that is most openly available is our so-called F2X libraries. We have 2 of them, a small one with 96 compounds and a large one with a little over 1,000 compounds. This is all published, so there is no IP connected to these libraries. You can just have them within the frame of a project or a collaboration if you prefer. Then we have access to the EU Open Screen libraries. The usage of EU Open Screen libraries is subject to some agreement with the EU Open Screen, which is another ERIC, as you know. So we are not totally free in giving out the EU Open Screen libraries. Then we have another one that's currently been developed with the Karlsruhe Institute of Technology. This is again some diverse, organic compound library. The beauty of that is that we have full control of the synthesis of all of these compounds and related compounds. That means we can easily work from there to improve the compounds that we find. The usage of that is not yet entirely defined. And then we have yet another one that we are developing or making available together with a facility in Brazil, the craft facility. And also there, we are not. I cannot yet say how this will be made available. At the moment, it's only available within the frame of a collaboration, but we also would like to make this more openly available. So in a nutshell, if you want to start, I would always suggest to use one of the F2X libraries, because we are totally free in using them. We have access to all. I mean, we essentially purchased all of the compounds. And this is all published in, I can send out the publication to anyone is interested. And, you don't even have to come to us to get access to this library. You can just purchase the compounds yourself if you like. Everything concerning this library is published.
Okay. Our next question, the computational platform is very interesting and seems to bring new things to the Instruct offer. So in terms of what they can do, are we also discussing sort of use of new sort of AI tools in both sort of prediction, but also in protein design? Are these the sorts of projects that people can work with you to develop?
Absolutely. I mean, if a piece of software that you require or that you need for your project is not available on one of these supercomputers, and if it's necessary, they will install this for you, or they will work with you to make this happen. That's exactly the idea of these. Of this project-based access. So in essence, you would write a project application to describing what you want to do and what you need, and they will help you make it happen, essentially.
Okay, all right. And then last question. So there's an activity in cryo-ET. Does this also then include the lamella production, the FibSem? Or are users expected to do that and transport their materials to the?
No, as far as I'm aware, it does include that. I would have to look at the detailed catalog. Maybe Pauline can answer that, but I think this is actually included.
Okay, all right. Well, I think I don't see any other questions right now. There's some information. From Pauline, which basically says, anybody can go and look at the catalog and get more information on the different centers, the facilities, and the platforms therein. So we invite you to do that. And I think we're basically at midday. So what I'll do is I'll finish the meeting and thank our speakers, Judith and Manfred, and look forward to the next Instruct webinar. So remember to sign up if you haven't already signed up, and then you'll actually get the emails which announce these events. Okay.
Thank you, Darren.
Thank you very much.
All right. Bye, everybody.
Well, thank you very much. Bye.
Thank you. Goodbye, everybody.